boys. Today we are here to demonstrate one more experiment for class 11th biology boys. This is meant for 6th January 2021. So previously we have done 2 experiments which come under major category 5 marks, and this also comes under 5 marks category. And this is very interesting experiment, as whenever you look at a leaf, green leaf, you will be preliminarily knowing, being biology, that the leaf is green or any part of the plant is green because of the presence of chlorophyll. Now the next question that arises in your mind is, will it be only chlorophyll or anything else? And sometimes it happens that you see yellow leaves, brown leaves, orange colored leaves, red colored leaves, similarly stem. So you get doubt, why is that color like that? Yellow, orange, red, brown, blue, sometimes mixture of that. You must have seen in crotons, mixture of colors will be there in the crotons leaf. So what for that? To understand that, we perform this experiment. So what is the aim of the experiment? The aim of the experiment is to separate plant pigments through a technique called paper chromatography. And what do you mean by this chromatography? Chrome means color, graphical representation. So writing based on the color, that is the meaning of chromatography, chrome and graphene. And we are thankful to this gentleman, Michael in 1906. He discovered this, so thanks to him for that. And what are the materials required? The second heading: fresh spinach leaves, already we have taken. Pestle and mortar, here is this. This is the pestle and mortar. So we have taken the leaves, and this is for grinding. And then funnels, of course. Wetman paper, this is the wetman paper, and then this is also called the chromatographic paper, filter paper, test tubes with a wide mouth, and cork, test tube with cork, measuring cylinder, pipette, micro pipette, or what we call it as capillary tubes, small sized capillary tubes. And of course, petri dish. So these are the materials we require. And the chemicals, to perform this experiment we need chemicals, because without these chemicals we can't do this. And what are those 2 chemicals that we require? Petroleum ether and acetone. And we require this in, we need to take it 50:6. So we have got this is the petroleum ether, and this one has got acetone. So we have got 2 chemicals. And of course, the procedure, how to do this. So step number 1, what is required is fresh spinach leaves, or any green leaves you can take, whichever is available to us. And after taking those fresh spinach leaves, now put some acetone and grind it. After grinding it, now filter. Now we just filter that into this flask. Once the filtrate is done, what is that you need to take? You have to take this wetman paper or paper chromatographic paper. Okay? And you take this micropipette or capillary tube, dip this micropipette in the leaf extract solution, liquid that we obtained from the leaf, and this is called point of loading. Now when you are taking this wetman paper, one edge is flat as you are seeing, another edge you have to cut in such a way that the sharp end will be there. So you take this micropipette or the capillary tube, dip it in that leaf liquid extracted from the leaf out of the grinding, and just touch that one spot. So touch once, dry it, go out in the sun, just wait for 30 seconds, come back, again put one more drop, just wait for 30 seconds, like this 15 to 20 times you have to do. Okay, 15 to 20 times you have to do. After doing that 15 to 20 applications, now you need to make a solution, and that solution is prepared out of ether and acetone in the ratio of 50:6 ml. So in that 50 is petroleum ether and 6 is the acetone, and then this solution is taken in a beaker like this, as we have taken, and put this, arrange the paper, chromatographic paper with the loaded liquid in such a way that it simply just go, but don't dip it, just the tip should touch like this, and leave it this arrangement for some time. Okay, just leave this for some time, and once you suspend that paper, chromatographic paper vertically, you can fix that with the help of pins and leave this for some time. Till how long you should leave? Till there is a raise in the carbon of the solvent. After you observe raise in the column of the solvent, you can see, take out the paper and again go out of the lab, stand in the sunlight and dry it. And now you can observe certain colors being raised. So that you can put it in the form of observations. So based on the procedure that you have done, you have to make observations, and the observations can be in a tabular form. So here, put a table, right, in the first column pigment, color of the band, and RF level. What is this RF level? That is, RF refers to retention front value, or it is also called relative front value. So this RF value is calculated by distance moved by the pigment and divided by distance moved by the solvent. So again I repeat, the formula for RF value is distance moved by the pigment divided by distance moved by the solvent. So when you calculate this, you get these RF values. So like you are seeing on the chromatographic paper, you will see that green color. You have put this loaded point, just above the loaded point you can see the green color. That is chlorophyll B. So you write chlorophyll B, pigment, color of the band, green. And what should be the RF value? 0.42. Next, you go to the next one. That is bluish green color you will observe on the paper chromatography. That is bluish green is chlorophyll A. Okay, and the RF value is 0.6. Then on the top you can see xanthophyll, yellow colored one. That should RF value is 0.72. And you can also see, because I am showing you here the one that we have done previously, you can see different colors. As I said, green, bluish yellow, also yellow, and yellow orange also you can see. That yellow orange is keratics, another type of pigments, and the RF value should be 0.95. And once you make this observations in the form of columns, then we go for the result. Based on the observations, you make result. As I said, either it can be present tense in one or two sentences, but of course I told you already, procedure and observation, since you are performing the experiment, it should be in the past tense and point wise. Do not write paragraph wise, write point wise. And what should be your conclusion or result? That with the help of paper chromatography, the pigments present in the given plant's leaf are separated and observed. This should be your result. And then once you make the result, then precautions. What are the precautions that you need to take are performing this experiment. First and foremost, leaves should be fresh. The fresh leaves, that's what we wrote, fresh spinach leaves. The leaves should be fresh, that is compulsory. Second aspect that you need to take care is the ratio between the two chemicals. Petroleum ether and acetone has to be 50:6. This is second precaution that you need to take. And the third one, most important again, this paper, chromatography paper which you are dipping after loading point, it should be a touch and go affair. It should not be dipped fully into the acetone ether category. So you need to fix the paper, chromatography paper properly. Because if you dip it, then the RF values will be of different one or the wrong ones. But the colors will be depicted properly on the paper, like as I displayed to you. You can see yellow color, orange color, blue, green color, and green color. All the colors you can see here. So, based on that, you need to take precautions. So, these are the various precautions that you need to take. And of course, the general precaution is when you are handling glass apparatus, you need to be careful. Don't drop like we are having the big mouths, wide mouths. No, you have got beakers. So these are to be handled carefully. And second, when you are handling the chemicals, be careful. Again, don't put chemicals on your body because they are very dangerous. So this is how you need to perform the experiment. At the end of it, as I said, whatever you are writing, it should be in the ruled page. And whatever tables you are forming, tabular observations and the diagram should be on the white paper. So observations, you write tabular form on the white paper and draw the diagrams. When you are drawing diagram, I have told you 4 things you must keep it in mind. 1, diagram should be scientific, it should be proportionate, the diagram should have an axis, and most importantly, value-wise, the diagram should be most beautiful. These are the 4 things you must keep it in your mind while drawing the diagram. Now, when you are drawing the diagram, this is the one that you must remember, loading point, and then based on the colors that we have listed, you need to label this. And of course, not to forget the solvent, because when we are writing here, look at this RF value, how to calculate? Distance moved by the pigment divided by distance moved by the solvent. So don't forget solvent. This is how we need to do this. Right? Thank you. We will see one more experiment in the next session.